Welcome friends. In last lecture, we have been discussed about the cell structure as well as the cell organelles and the outline of the functions of various organelles. Today, we will start with the ultrastructure and functions of the plasma membrane. So, plasma membrane is nothing but the limiting membrane or the outer covering that is in the form of the unit of a specialized kinds of the biomolecules, and because of that will be limiting membrane is formed around the cell, each and every cell of the organisms, okay, as well either prokaryotic or the eukaryotic. There is a outer membrane is there which will be protecting the cell from the external environment, and that will be maintaining the structure of cell. And so that plasma membrane is one of the important structure which is helpful to protect the cell from the external environment. So according to the structure of the plasma membrane, it cannot be seen by the light microscopes. The workers have been observed it under the electron microscope, and as the cell membrane around the cell periphery, around the outer periphery, as well as whatever the cell organelles, the different organelles we have been studied, there are these organelles like endoplasmic reticulum, mitochondria, Golgi complex, lysosomes, ribosomes, centrioles, all the cell organelles having the membrane. That is these are also the membrane bound structures. These are also the laminar structure, trilaminar structure of the cell organelles also itself, as well as the plasma membrane of the or the unit membrane of the outer periphery of the cell above the cytoplasm. There is presence of the plasma membrane. So according to the study of plasma membrane, that will be protect the protoplasm as well as the cytoplasm of the cell, and that forms the boundary of the cell, as well as that plasma membrane is very thin and elastic in nature, and it is permeable to the it is not the completely bound to the cell around the cell, but it is having a specialized pores are there, and that pores are permeable. That because of that pores of the alternate pores on the plasma membrane, these are allowing the specific substances to the cell, as well as for the exchange of the materials into the cell, so that under the electron microscopes, observers or scientists have been observed the plasma membrane, that is a limiting membrane. And if we study about the that is the structure, the outline of the structure, there is a bilayer structure of the plasma membrane is there. There is a bilayer structure. That bilayer is of the protein and lipid two layers. That is the proteins are situated at the outer periphery of the to the external environment of the cell, as well as inside the cell structure. For example, suppose if we see the plasma membrane, that at the outer periphery there is a proteins are situated, as well as inner margin of the cell, integral of the margin of the cell above the cytoplasm, there is also the protein layer, as like here. And then in between the protein bilayer, there is presence of the that is the monolayer around the outer exterior as well as the inner surface of the cell, and then inside that there is a presence of the bilayer of lipid structures. Okay, that lipids are especially phospholipids, and it has been invented by the observations of the various scientists. But first of all, in 1935, there is Danieli Dawson, scientist Danieli Dawson, first of all proposed the membrane, that is the unit membrane of the cells. But there is some exception in his observations, that is the he have been proposed some indirect evidences. There is no clear picture about the at the time of the observations, initial observations of the Danieli and Dawson that workers. Then after that, in 1959, there is Robertson have been found the structure of the appearance of the trilaminar structure, that is the protein, lipid bilayer and the protein structure. And so that Robertson have been first of all proposed the plasma membrane structure, the actual trilaminar structure of the plasma membrane. So the plasma membrane is also present, that unit membrane has been formed into the plasma membrane as well as the endoplasmic reticulum, ribosomes, mitochondria, all the cell organelles having or covered by the plasma membrane, or they are also the membranous. So according to the plasma membrane structure, that varies in various cells, that is most of the cells having the size of the or thickness of the plasma membrane is around 100 to the 250 angstroms in thickness. In general, there is most of the cells having the structures or the thickness of the plasma membrane around up to the 100 to 250 angstrom units. So plasma membrane in most of the cells there is present that the metabolic activities of the cell, because of that plasma membrane there is a metabolic activities of the cells have been formed or cells have been takes place. So that lipids are the molecules in a chain. There is a fatty acid chains are there, as well as there is a two layers of the lipids are there. First layer at the outer exterior surface and the interior of the cell. These are two layers are there. So according these are interact with the protein layers, exterior and the interior margin of the cell, and then because of the van der Waals force there is a bonding of the hydrogen bonding of the in between the lipids and protein bilayer, and so that they are interact with each other, and so that there is a formation of the plasma membrane is there. So if we study about the this chain of the lipids that remain both lipid layer joined with each other, and then they are joined with each other by the hydrophilic head regions and the hydrophobic tail regions. These are the fatty acid chains. If we observe that there is a structure of the tail region, there is hydrophobic tail regions are there and the hydrophilic head regions are there, and above that there is a protein monolayers are situated. There is a protein monolayers are situated. So first of all, scientists have been proposed the actual structure, trilaminar structure of the plasma membrane, and as far as concerning the structure of plasma membrane, if we see that is the together they are formed the van der Waals force at the protein and the lipid structures, and so they are interact with each other, and there is a fixed to the van der Waals. Because of the van der Waals force there is a membrane is fixed with the glycoproteins as well as the lipoproteins, glycolipids and the glycoproteins, so that there is lipids and proteins, means lipoproteinous structure of the plasma membrane or the unit membrane have been takes place. So most of the cells are made by the whatever the lipid bilayer, that is of the phospholipids are used. First of all, many scientists have been proposed that there is not clear picture or evidences where there is the actual lipid bilayer is of which kind of the lipids are situated in the formation of the plasma membrane. So after the studies of the Danieli Dawson as well as the Robertsons, they have been proposed the trilaminar structure, that is one monolayer at the exterior and one monolayer at the inner margin of the cell, and then in between that there is a presence of the head and tail region of the glycolipids. And then so if we see the structure of the plasma membrane, that is a trilaminar structure. So here is the clear picture, that is cell is maintaining the metabolism as well as the all the activities into the water. So if you observe the structure of the plasma membrane, that is there is water is situated and the lipid layer is with the two fatty acid chains are attached to the head portion of the structure of the plasma membrane. So mostly there is phospholipids are used that formed by the into the plasma membrane. These are the lecithin mostly and the cephalin. These are the two kinds of the structures or the phospholipids which are used to form the plasma membrane, as well as the proteins are there, and then there are formation of the plasma membrane is there. If you observe the tail head, that is, tails are the hydrophilic and the heads are the hydrophobic. So that tails are at the exterior, they are present, tails are hydrophobic and the heads are hydrophilic, means above outside the cell there is a water medium is there, and inside the cell there is also the gelatinous fluid like jelly substance in the cytoplasm is there, and so that there is also the watery substances are there. There is various droplets of the biomolecules, as well as the cell organelle is also the bounded, and so that membranes are forming the compartments of the cell. As far as concerning the all the cell organelle is also consisting with the plasma membrane, so that these plasma membrane containing membrane organelles of the cell are forming the different compartments of the cell into the cytoplasm. So according to the structure, trilaminar structure, if you observe that tail are hydrophobic, and head are hydrophobic, as well as there is a phosphate group is attached to the choline, that is the protein, and the cholesterol is also in the form of the steroids. There is a steroidal structures are also present into the in some form, and there is presence of the cholesterol is there, and then lipid, one chain of lipid and second chain of lipid with the, that is called as the lecithin, and the glycerol is there. So according to the structure of the cells, if we observe the there is a structure of the lipid, that is the head are the water loving or polar regions, and the tails are the water hating, that is the non-polar regions of the lipids are there. So that there is a mutually proteins and lipid are forming the trilaminar structure, that is on both the sides there is a proteins are there, and inside that lipid bilayer is there. So according to the chemical composition of the plasma membrane, if we observe, the mostly proteins and lipids are small in small percentage. There is presence of the carbohydrates are also there, and so that they are forming the glycoproteins as well as the glycolipids. So according to the structure of the plasma membrane, if we observe, the main structure of the plasma is formed by plasma membrane is formed by the lipid bilayer as well as the two monolayers of the proteins. But in some quantity there is carbohydrates are also present, and if you observe this structure, the how much amount of the approximately there is a it has been observed that there is approximately 52% of the proteins are there. 52% of proteins are there of the formation of the plasma membrane thing, and as well as there is presence of the up to 40% there is lipids are there. Okay, they may be there are, these are the phospholipids. Okay, lecithin, either lecithin or the cephalin. Okay, so 52% proteins, 40% lipids, and it is the average of the up to the, it is observed that in different cells there is 1 to 10% of the carbohydrates, but it has been imagined that 52% of the protein, 40% of the lipids, and the 8% of the carbohydrates are present at the formation of the plasma membrane. So that is the chemical composition. If you observe, there is a different kind of the biomolecules or the macromolecules are present for the formation of the plasma membrane. So according to the various different cells, there is a presence of plasma membrane in the form of the their ratio of the plasma membrane of the proteins and lipids. In some cells there is 80:20 protein and lipid bilayer is there. In some cells there is 20:80 concentration of the proteins and lipids are there in some cells, and most of the cells are are consisting with the 50-50 in nature. 50% of the lipids and the 50% of the proteins are there. So according to the carbohydrates, there is some they have been some synthesis. There is a Bale scientist have been observed, that is, he have been proposed that there may be up to 5% of the carbohydrates are situated. He have been proposed his theory into the 1962, and he have been observed that there is maybe 5% of the carbohydrates are there, but the lipids are present into the plasma membrane, and in the form of the carbohydrates there is hexose and the hexamine, as well as the sialic acid, that is in the form of the carbohydrates. There is presence of the carbohydrates in the form of the hexase, sialic acid, as well as the hexamine, and so that these are the helpful for the binding with the proteins and lipids, and they are form the biochemical reaction. They are forming the glycoproteins as well as the glycolipids, and so that there is a structure is formed. And then there is lipids are there. Plasma membrane containing 20 to 40% of lipids in different cells. As far as concerning the very specialized kinds of the cells, that phosphorylates, as earlier we have been discussed, that is the lecithin and cephalin are the major phospholipids which are present for the formation of the structure of the plasma membrane. So they are forming the phospholipids, glycolipids, and the steroids, that molecules, that is basically these are contains the head, that is the head regions, that is water soluble, and the tail regions, that is the fatty acid states, which are against the water. These are non-polar regions. So according to the structure of the proteins, there are specialized kinds of the proteins are there. If you observe the protein structure first of all, then there is into the formation of the plasma membrane, or the structure maintenance of the plasma membrane, there is presence of three kinds of the proteins are there. There is structural proteins as well as enzymes. Enzymes are nothing but the proteins, as we say. Okay, then structural proteins, enzymes, and the carrier proteins, we can say. There are three kinds of the proteins are there, and most of the role is played by the structural proteins, because they are at the around, and as well as they are situated at the inner margin of the inner surface of the cell, as well as the outer exterior periphery of the cell. And these structural proteins are having or consisting with the various enzymes also, and they are protecting the cell from the different kind of the unwanted materials to the exterior to the cell as well as interior to the cell. Then there are many kind of the enzymes which are helpful to protect the cell from the outside medium. Okay, there are different kind of the enzymes are there, as well as there is presence of the specialized carrier proteins, and that carrier proteins are nothing but the permeables. These are called as the permeables because they are permit to the transport of the different kind of the biomolecules or the macromolecules into the cell or from to the cell. That biomolecules or the debris or etc. that will be passed through the pores of the plasma membrane, because plasma membrane is a continuous membrane. There is alternately there is presence of the pores are there, and the pores are also provided with the protein and lipid structures, and so that there is a permeability to the specialized kinds of the substances which are required or unwanted to the cell, and because of that transport of the various substances from the cell and into the cell because of the carrier protein, that role is played by the carrier protein. So that there is specialized kinds of the three types of the proteins are there, and that proteins are structural proteins, enzymes, and the carrier proteins. So according to the we have been observed about the chemical composition, then there is the proteins are there. That carrier proteins are in the form of the specific integral peripheral proteins, and that integral peripheral proteins are integrated at the exterior with the lipid bilayer. They are integral proteins that integrated, and peripheral proteins, and integral proteins are forming the layer and attached to the, in addition to the integral proteins, they possess the hydrophilic amino acid macromolecules, because proteins are made by the macromolecules, that is the amino acids. So these are hydrophilic amino acid chains into the proteins are there, that is structural proteins, enzymes, as well as the carrier proteins. These are the hydrophilic amino acid macromolecules are made the specialized kinds of the structural enzymes as well as the carrier proteins of the cell membrane. Okay, so according to the amino acid macromolecules that exposed to the surface of the molecule of the cell membrane, as well as that connection with the there will be that connection with the ions, various ions, as well as the hormones, as well as the neurotransmitters. Okay, transmission takes place at the time of the various cell activities, and then there is a different kind of the antigens are also alive. So that formation of the proteins takes place into the formation of the plasma membrane. Then if we observe, there is lipid chain first, lipid chain second, and the cholesterol is there, and then as well as there is a glycerol in the form of the glycoprotein and glycolipid structure. There is glycerol is present, as well as the phosphate group is attached to the lipid and protein bilayer, so that there is lipid, especially lipid bilayer, and that phosphate is nothing but the mineral, and that phosphate is attached to the formation of the structure of the hydrophilic and hydrophobic nature of the lipid bilayer, and so that there is lipids are present. So that is the chemical composition of the plasma membrane structure, and that is approximately up to the 52%. It has been imagined that 52% of the protein, 40%, approximately 40%, 20 to 40%, different to different cells. Okay, so 20 to 40% as well as up to the 50% largely proteins are present, and that proteins are consisting with the structural enzymes as well as the carrier proteins. 20 to 40% there are phospholipids are there in the form of the cephalin or the lecithin. Okay, then these are 20 to 40%, and up to 5 to 8% there is presence of the carbohydrates are there. They are forming the or making the glycolipids and glycoproteins. Okay, so according to the functions of the unit membrane that is forming that is discussed by the various workers, that is called as the unit membrane, that will be forming the specific limiting membrane to the cell, forming the limiting membrane to the cell. And so that if we observe about the functions of the plasma membrane or the unit membrane, that membranes are make the compartments inside as well as the outside, exterior to the cell as well as the inside the cell, as we discussed into the observations or study about the outline study of the various cell organelles into the pro and eukaryotic cell. So there is especially eukaryotic cell, there is various organelles are there. So there are membranes are forming the compartments into the cell interior. Okay, then cell membranes are interact with the connections with other that cells, and they are forming the tissue. Okay, with other cells there is formation of the structures of the tissue have been takes place, and then that at the time of the embryonic development, at the time of the development or the organogenesis, at that time cells are forming or interaction with the other membrane, other cells, and they are formed on the basement membrane to interact with the other cells, and they are forming the tissue. So that is the one kind of the characteristic or the function of the plasma membrane. And cell membrane protect the cell from intake of the hazardous particles inside the cell. Okay, that membrane is also protect the intake of the inside the cell, that will not allow the hazardous materials to get inside the cell. As well as another characteristic, that cell membrane or plasma membrane is play the major role to the response to the signals, to the, as we earlier said, that various ions, hormones, neurotransmitters, antigens, they are allowed by the selective permeability by the cell membrane. There is permeable carrier proteins are there, these are also called as the permeables, and so that these carrier proteins are allowing the whatever the required as well as the unwanted substances from the cell or into the cell, and so that these carrier proteins are play the major role into the plasma membrane. So that is the one most important characteristic, that signals, whatever the plasma membrane is play the major role into the signals recovered by some specific agents like the neurotransmitters, antigens, hormones, ions, etc. Okay, then it also play the role as it protect the cell from the losing the various macromolecules from the cell interior, into the cytoplasm. Okay, so into the cytoplasm there is various macromolecules are there, various biomolecules are there, as well as the jelly, that skeleton is there of the cell of the cytoplasm, and so that all these with organelles and the all structure of the cell or the skeleton of the cell, that will be protected by the plasma membrane. Okay, so plasma membrane also protecting the or maintaining the integrity of the cell. It will be maintaining the integrity of the cell. So according to the some scientists, they have been demonstrated that that in their studies, that membrane, as like the Langmuir, that have been suggested that is formed by the phospholipids, as well as the various scientists, that is the Garter and Grander. They have been observed that first of all, the water loving and water eating, that lipid molecules are present inside the plasma. That these were the initial studies of the plasma membrane. At that time, Grant and Glender, that scientists have been proposed the lipid hydrophilic and hydrophobic nature of the lipid is present into the plasma membrane, as well as that inner and outer environment, there is aqueous structures of the hydrophilic and hydrophobic lipid layer, as well as there is heads are hydrophilic and the tails are hydrophobic. So as we study about the concept of plasma membrane, that concept of plasma membrane is formed or the majorly based on the that in 1953 Robertson have been proposed that or observed to suggest the Danieli Dawson in a right evidences. In a right evidences, he have been proposed a Danieli Dawson model in a right evidences. By the right evidences, he have been proposed there is a trilaminar structure of the plasma membrane. So that trilaminar arrangement of the plasma membrane in the eukaryotic and prokaryotic cells, there is presence of the specialized kinds of the proteins and lipid bilayers are there, and that protecting that, as the Robertson shown that under the electron microscope, he have been observed that there is a whatever the structure of formation of the structure of plasma membrane, there is specialized observations have been proposed by the into the Robertson structure of plasma membrane. So as we see, as the exterior, suppose there is exterior of the cell, there is double layered structure of the plasma membrane. At the exterior, there is presence of the proteins, that is up to the 20 angstrom in diameter, okay, in the thickness, okay. Then there is presence of the lipid bilayer, that is up to the 35 angstrom, and the inner margin of the cell, there is another monolayer of the proteins is situated, that is also the 20 angstroms, that is also 20 angstroms. So mutually there is the thickness of the plasma membrane, that is the there is some spaces there also present, but the lipid and protein trilaminar structure, that is up to 75 angstrom units. Its thickness is up to the 75 angstrom units is the thickness of the plasma membrane. So it was considered the trilaminar structure of the plasma membrane, that is lipid and protein bilayers, and whatever the head structures, that is hydrophilic or the polar structure of the head, that is up to the 5 angstrom, and with total unit of the hydrophobic tails and hydrophilic heads mutually, that is up to the 35 angstrom, and the 20 angstrom at the exterior and the inner margin of the cell, 20 angstrom each layer of the proteins is situated, and so that there is total thickness of the membrane have been 75 angstrom, and so that Robertson first proposed that in 1953 and up to the 1959 he have been proposed that there is a trilaminar structure of the plasma membrane, that under the electron microscope or the X-ray diffraction studies, there is further studies have been that arrangement into the light arrangement onto the lipid bilayer, there is presence of the major class of lipids, that is characteristically hydrophilic and hydrophobic natured lipid is there, and that structure is whatever the chains of that structure of the lipid is as like this fatty acid chains, which are consisting with the structure of the specialized kinds of the structure of the with carbohydrates, that is the formation of the glycerol is there. There is glycerol structure is there, and that glycerol structure will be attached with the fatty acid chains, and there is formation of the glycolipid is there, and that glycolipid is again formed into the head portion also. It is the tail portion as well as it is the head portion, that is phosphoglyceride. There is the glyceride structure is formed at the lipid structure, and there is combination of the phosphate and the lipid structures, lipid biomolecule, and so that there is the formation of the phosphoglyceride, and so we are called, there are phospholipids are there into the formation of the, they are play the major role into the formation of the plasma membrane. So it is the structure of the head and tail portion of the plasma membranes, and there is hydrophilic head and the hydrophobic tail portion is present at the plasma membrane. So phospholipid molecules are between the protein molecules that form the lipid bilayer and the permeable water and natural non-polar molecular structures, they are allowed by by this biological membrane, that is the protein and lipid combined with the polar structures. They are facilitated to the transport of the various molecules or substances, transport of the various molecules or biomolecules and the macromolecules or other substances, that is ions, hormones, neurotransmitters, that will be allowed by the lipid and protein bilayer, and so that that facilitate the transport across the membrane, and so that cell will be maintaining the life, and so cell may be maintaining the life. And like this, there is a, first of all, the various scientists have been proposed the structures, what is how the structure of the plasma membrane, how the plasma membrane formed into the cells, and that have been proposed by the various theories or various proposed by the various models. And in next lecture we will discuss about the how the Danieli Dawson, first of all, in 1935 he have been observed the bilayer structure of the plasma membrane, but there is no right evidences he have been proposed, but he have been proposed the outline of the plasma membrane structure, that is how the plasma membrane have been formed. So he have been we will study in next lecture about the Danieli Dawson model of the plasma membrane, as well as the another model, that is the fluid mosaic model of the plasma membrane. So आज के लिए हम यही रुकते हैं, और in next lecture we will discuss about the two models that proposed by the various scientists, Danieli Dawson and other scientists, that is models of the plasma membrane. So आज के लिए हम यही रुकते हैं, और कल हम स्पेशलाइज्ड मेम्ब्रेन स्ट्रक्चर हम स्टडी करेंगे. थैंक्स.